Welcome to an exciting session on assisted gene flow today. I am very excited to introduce our first speaker, Dr. Nick Kooyers, who is here from the University of Louisiana, Lafayette. And the title of his talk is Consequences of Assisted Gene Flow on Threatened Annual Monkeyflower Populations.
Ooh, it's a little high. There we go. Thanks, everybody. Thanks, Elsa, for the introduction there as well, too. So one thing I probably don't have to convince this audience of is that many plant populations are threatened by climate change. And I know this fairly well from my own research because I just got done doing a large review paper on all kinds of herbaceous model systems and really summarizing the evidence from all those different systems. And what we found from those systems that are not the things that you typically would think would be threatened, they're widespread, common species for the most part, is all of those species still are maladapted or have declining populations somewhere in their range. And all of them, plasticity does not seem to be sufficient to be able to recover fitness in those threatened populations. And all of those species have dispersal that's limited between different populations too. So we really need to think about what we need to do to be able to address this dilemma here. Assisted gene flow is one answer that we're going to be talking about in this session today. Assisted gene flow as a basic answer is this human-assisted migration of populations within their species range in order to facilitate adaptation to local conditions or increase genetic diversity in target populations. In principle, this looks like removing populations often from hotter, drier regions within their species range to threatened populations further north in what has been historically colder environments. I like to think of this not as migrating to new areas, but instead this is basically accelerating gene flow. These species are already exchanging genes between these populations. We are simply accelerating that process. Now, assisted gene flow is controversial, and that's probably the one thing that you've heard about assisted gene flow. And one of the reasons for that is because it's relying on this evolutionary model of evolutionary rescue, where if we have some threatened population over here, population growth rates are typically below 1, below replacement level. When we have assisted gene flow actually occurring, we think that we might get some spike of maladaptation from the introduction of some maladaptive alleles early, but then any adaptive alleles are going to get raised in the population and we're going to lose those maladaptive alleles. So there's concerns about initial maladaptation, and there's concerns about if it works too well when we replace native biodiversity in those different areas. So a big question is, how does introducing new alleles actually impact natural populations? And there's very few case studies of assisted migration out there, especially in a controlled context. Really what we'd want to know in a controlled experiment is, with assisted migration, how are those alleles introduced from that source population? How fast does the changes in phenotype occur? And does this actually alter fitness in those threatened populations? Okay, so we can kick some expectations for a successful assisted gene flow. And in these controlled conditions, we're going to have baseline data collected pre-assisted. We're going to have baseline data collected pre-assisted gene flow, data collected post-assisted gene flow, and we're going to have control populations where we don't introduce those new source populations, and we're going to have populations where we induce those source populations from hotter, drier regions. Okay? And what we might think that we'd see is in control populations, pre-versus post-assisted gene flow, we might not see a difference in fitness, but in the assisted gene flow populations, we hope that we see an increase post-assisted gene flow relative to the pre-assisted gene flow populations. You could think about the same thing for phenotypes where we see a shift in phenotype, but only in those post-assisted gene flow populations in our treatment, not in our control. And finally, you could think for genetics in this, that if we're looking at the donor private alleles, so alleles that are private to that source population that are not found in our threatened populations beforehand, we would like to see that you would actually see some increase in those private alleles in the populations that were the treatment populations relative to the control. I have this over here to remind us that those alleles that we introduce in the AGF populations are likely going to spread post-assisted gene flow even into our control populations over time. Okay, so the species I'm working with here is Mimulus guttatus, or Erythranthera guttata, if you prefer that name. And it's been used as a long time for a model system for ecology and evolution, largely because it has this common species that has a really wide geographic range, all the way from central Mexico to central Alaska, from sea level to 11,000 feet. It exists over all these different habitats, and it's a model for local adaptation. It has a great molecular toolbox with a couple of great sequenced genomes, many resequenced genomes in it, a long history of ecological experiments, and it has really well documented for some of its trade-offs, particularly those between reproduction and chemical and physical defenses. All right? The one thing you need to know about guttatus in this particular monkeyflower is that it's very much limited by water availability, especially in the annual populations that I look at. So populations start their growing season essentially with spring or winter rains or with spring or winter snowmelt, and they end their growing seasons essentially in that hot, dry summer that we're so familiar with here in this Mediterranean climate. So this is what they look like at the end of the growing season here. Okay, most of you probably are familiar with the common monkeyflower as a ditch plant. It's all over the place in California. It's super common. So you're like, why are we doing assisted gene flow here? Well, in fact, in some populations, it is not doing so well. And those are the populations I study in the Central Oregon Cascades. I originally started studying this plant as a model for local adaptation. But during my postdoc, one of the first things I did was I went and did a provenance study where I planted low and high elevation populations from California and low and high elevation populations from Oregon into low and high elevation sites in Oregon. I expected to find some lovely local adaptation and get some genetic basis of that. Instead, what I found was that the California populations from low elevation did better than the low elevation Oregon populations in the low elevation site, and that the populations from high elevation in California did better than the native Oregon high elevation populations at the high elevation site in Oregon. This is a classic signal of an adaptation lag. Thus, these populations from California that are adapted to these earlier growing seasons, and this growing season has shifted dramatically in these populations. For a 6 to 8 week growing season, it shifted 2 weeks earlier, closer to the California growing seasons. So these populations, in fact, can represent this kind of canary in a coal mine. Even in a common species, we still see that these populations are threatened. When I started my faculty position 8 years ago, I decided that I wanted to sit in these high alpine meadows for most of my existence, and I'm going to continue to do that. So I started tracking 12 of these populations in these high elevation sites in Central Oregon. Every year I'd go out to these sites and I'd run transects across them. We count seeds from about 50 to 100 plants per population to get fitness measures. And we look at offspring phenotypes when we brought these seeds back into greenhouses to grow them in common gardens. And then for each one of those plants, we would then collect whole genome sequencing data. We did this for a subset, for 24 individuals per population per year. These are what those populations actually look like. Okay? So, the general result of that was that after the first couple of years, I saw one of the populations go extirpated, another population that dropped below 50 individuals in that single year too. So 1/12 of my populations went completely extinct during that time. They subsequently came back later with the seed bank a couple of years later after that. But what I decided to do was I decided to make this into a landscape scale assisted gene flow experiment. You notice that those 12 populations are kind of grouped into 4 groups of 3 populations. Well, in each one of these groups of 3 populations, like these purples down here, I planted one that was control where I threw out 200 native seeds from that population. In a second population, I planted California F1 crosses, a low elevation and high elevation plant that I threw in there. Why those populations? Well, those perform the best in common garden studies. Then I also planted out seedlings in the third population from that same cross right here, but about 1/10 the number of those different seedlings up there, in case germination might have been a barrier to assisted gene flow. And I kept tracking those same fitness measures, the same phenotype measures, and the same genomes over time. Okay, so that was in 2018. We had 3 baseline years pre-assisted gene flow, and now we're through 2025, and that's the day I'm going to show you today. So this is 8 years of work going into this particular experiment right here. And I want to remind you that we expect relatively limited changes within the first generations following assisted gene flow, especially from those evolutionary rescue models. These are relatively large annual populations when they have good years, and that means introducing alleles into them, they're likely going to be at low frequency to start out. Okay, so what do we find? Well, the first thing that we found, and this has happened after 3 years is the first kind of data that we had on this, is that if we did assisted gene flow using seeds, we increased fitness. So you see floral abundance on this axis over here, and you can see there's a slight increase pre-assisted gene or post-assisted gene flow relative to pre-assisted gene flow. That's in the seeds. We see no change in the seedlings, and we see less of a change in the control populations. So relative to the control populations, we saw about a 20% increase in floral abundance in those seeded populations. If we did this using seeds, the seed production instead of floral abundance, we found a similar 20% increase, but it wasn't statistically significant over those different 5 years that we have there. So we find some effect generally of this, of increased fitness within these metapopulations. However, there's a lot of variation there, so we decided to break it into the different blocks, those 4 different blocks of 3 populations. And when we did that, we see really strong effects in one of the populations. Here's, again, the seeds population for the treatment, a big jump, reasonably small jump in the seedlings, and you can see that post-assisted gene flow actually has lower fitness than pre-assisted gene flow in our control population there. However, we also see something else, which is creating that variation within that full metapopulation plot. We see that one of our populations actually had male adaptation following assisted gene flow. Again, in the seeded populations, we see that they're relatively even pre versus post-assisted gene flow, but there was a jump in the control population and a jump in the seedling population. Thus, that seeded population is doing relatively worse than the control population post-assisted gene flow. So this corresponds pretty well to what we expected from evolutionary rescue models from assisted gene flow. Okay, there's also strong differences between years. So the years we planted out seeds in the first place was 2021. What you can see is this is a graph that essentially shows the difference relative to the control population. So positive numbers means increases in flower production relative to the control population, and negative numbers means decreases. Okay, so that's 2021, and here's 2022 and 2023. We can see that almost all the populations increased relative to the control populations in 2022, but the major ones that had the increase are these OWC and RRM, had decent jumps both in 2022 and a lesser jump in 2023 relative to the control populations. Whereas at BMP and MTD, those are our male-adapted populations. You see in 2023, that's where they dropped off relative to the other populations there. So there's differences between years and how well assisted gene flow worked. This largely had to do with the different conditions during those years. 2021 had a really intense heat dome, and 2023 was dry, but 2022 was historically pretty wet. Okay, so what I'm telling you here is our assisted gene flow was variable among populations, where we had some populations where it worked in, and some populations where it did not. We saw that the seedling treatment was more effective than the seedling treatment, and we saw that there was variation going on between years. So we know that the conditions that we have this assisted gene flow treatment in, it's actually going to depend on those conditions in the particular years that we're doing this experiment, okay? All right, so thinking about phenotypes then, what traits are actually underlying that fitness variation, and have populations evolved more towards the California source populations? We did a gigantic common garden experiment with 6 years of data, 3 pre and 3 post assisted gene flow, all 12 populations and about 24 lines per population. Courtney Patterson was the tech in charge of this particular experiment right there, and this is what it looks like. We did a bunch of phenology measures, morphology measures, defenses. This is a heck of an effort by Courtney. This is thousands of plants right here, all for these graphs. And what we see is small effects on that where the seeds population had lower or earlier flowering time. We see that they had less trichomes, and we didn't see very much difference in defense. But this is the entire metapopulation. What happens if we bring this down just to those populations where we know assisted gene flow actually worked in? In those cases, what we see is that we see a strong, relative increase in flowering time, earlier flowering, we see less trichomes, and we see lower defenses in those treatment sites that have increased fitness there. So we have some traits that actually underlie assisted gene flow. If you compare these to the source California populations, both of these populations are earlier flowering, but the California populations are much larger with faster growth rates and low defenses. compared to the organ populations. What you can see is it's very clear that you have those same kind of phenotypes that are popping up within these particular populations that had changes in fitness. Okay, so we do see some changes in phenotype. How about those genetic changes? We have whole genome sequences, and we're most interested in is integration genome-wide, or do we lose native biodiversity here? These are the important questions that we wanted to ask that were the concerns about assisted gene flow in the first place. So we took a bunch of samples pre-assisted gene flow, a bunch of post-assisted gene flow, as well as the California parents that we have in there, and sequenced whole genomes. Then we extracted private alleles, so single nucleotide polymorphisms that are private only to the California population or at high frequency within that population. And we track these. And what we found, and we had excellent depth across there, just statistics if you like genomics. What we find is in that particular block where we had those increases in fitness, we see that we have greater integration, particularly in 2021, in our seeds and seedling populations relative to our control populations. We see the same thing. We have integration in the populations that had a decrease in fitness with assisted gene flow, mainly again in 2021, right after assisted gene flow. And if you go back to looking at that populations that had the increase in fitness, we can look at the frequency of those private alleles in California, and we can see that they're all generally below 10% within the population. And those private alleles, the frequency in the populations kind of changed between 2021 and 2022. This suggests that there's integration genome-wide here, and it differs in between different groups, but it's still occurring at really low frequencies. To take a look a little bit more at the broader population scheme here and see if we lost genetic diversity, we just did a broad PCA just with the California populations and all of these Oregon populations. And what we find when you do the PCA generally is we see California populations grouping over here. Here's our, the red is the Bell parent, the pink is F1s. These are all the Oregon samples over here. What we find is the main factor differentiating these populations is that PC on PC1 here. If we zoom in a little bit, these are the Oregon populations over all of the years. So that's just this right there, basically. So you see no difference really between years, including pre versus post-assisted gene flow in those Oregon populations. If we break it down further and look at box plots like before, what you can see is the only one where you really see significant integration in, particularly in the seeds population, is in the population block that had that increase in fitness. And you can see it's moving in the direction, so a positive PC1, towards more California alleles within that population. So we do see the signal of assisted gene flow in these populations, and there's very little native variation that has been lost in these populations despite that assisted gene flow that's there. So it's kind of alleviating our concerns. Okay, so the takeaways from this example of assisted gene flow right here is that assisted gene flow can occur extremely rapidly and that we have certain logistical considerations, for instance, introducing seeds that can stay in the seed bank for multiple years, that can be useful. We see multidimensional phenotypic change, where it's flowering time and defense, and these correlations make some difference in how we see assisted gene flow working. What I didn't tell you about is the direction that flowering time actually moves makes it predictable which populations are actually going to increase versus decrease in fitness right away. Okay? We see integration genome-wide, and we see very little loss of native biodiversity here. But I want to say that best practices for assisted gene flow are still needed. There's a lot of different logistical considerations that we need here, and we need to figure out how to best pick source and target populations. But I'll leave you with that and take questions. Before I do that, though, I want to thank a lot of people, everyone in my lab, that helped out with this project, particularly Donna Hendricks, this is her master's thesis for part of it, and Fernando Hernandez, who is our lead bioinformatician on this. I also want to thank the Forest Service for permits for this because they believed in this project enough to give us permits to do it. So thank you.
So thank you, Nick, for that great talk. You actually used all of your time, so we might have to save your questions until the panel at the end. So for those of you who've looked carefully at the schedule, you'll see that we have 4 related talks on assisted gene flow and then a 20-minute panel at the end, so lots of time for discussion. So I encourage you to ask questions after the next couple of talks that are specific to the talks themselves, and then save those really meaty, juicy questions about assisted gene flow to the end. All right. So it is my great pleasure to introduce our second speaker, Dr. Brandy Quarles-Chidingaya, who is here from UC Davis. And the title of her talk is Assisted Gene Flow, Not Assisted Migration, May Facilitate High Elevation Population Persistence for Mountain Jewelflowers.
Thank you. Thank you for everyone for coming. I'm just going to jump right in. So we know that climate change is impacting climate globally, but especially at high elevations, we're seeing increased warming and reduced snowpack and earlier snowmelt. We also are seeing changes in our seasonal timings, where our springs are arriving earlier and summers are lasting longer. And so the big question is, how are plants going to respond to these changes? They can adapt genetically in place. They can disperse spatially to try to track these changes, or unfortunately go extinct. And of course, there's a lot of complexity built into this, but I find it a useful framework to work from. And so when we consider a high elevation plant currently growing at high elevation, after climate change, since it's historically adapted to these high elevation conditions, it may not be able to adapt to the new changes in climate due to the low genetic variation. Whereas a plant that used to occur at low elevation may be pre-adapted to these warming conditions at higher elevations, and so there's potential for upslope migration. But there's complexity here. Of course, with different elevations comes different seasons, different life histories, especially in Mediterranean climates. And so at low elevations, plants have to deal with hot and dry summers, so their germination is usually induced by fall rains, and they have winter or spring growth. Whereas at high elevations, plants have to contend with high snowpack from late fall through spring, and so their germination is usually induced by spring snowmelt, and so they have summer growth. So these plants are growing at very different times of year, and also because of the low growth seasons at high elevation, plants usually grow as biennials, whereas at low elevation, they have more time to grow, and so they can sometimes grow as annuals, sometimes as biennials. So there's even more complexity there. And so if we consider performance at high elevation during the hot, dry summer, we may see that our high elevation plants may not survive very well, but our low elevation plants may perform quite well in those warmer conditions at high elevation. But when we consider the winters, even though it's warming at high elevations, they're still getting snow, and low elevation plants aren't used to snow. And so if we consider germination phase, high elevation plants may know not to germinate until the snow is gone, but a low elevation plant might try to germinate at the wrong time, which could cause a pretty low seedling establishment. And then if we consider growth into the second year, where even though high elevation plants may not survive very well during their first summer, maybe they'll be able to handle that second winter better because they're used to it, whereas low elevation plants may be too tall, and so they're not gonna maybe get protected by snowpack and maybe have low survival. And so that brings me to my research questions. First, are local high elevation populations showing signs of adaptational lag due to recent climate change? And consequently, do plants from low elevations perform better than the local plants at high elevation? And will plants from hotter origins survive summers better at high elevation? So is there a seasonal dependency to this difference in survival between plants from different elevations? And so will plants from cooler origins survive winters better? And then finally, if we actually try to think about assisted gene flow potentially, will crosses made between plants of cool and hot origins be the best option? Okay, so I've been working with Streptanthus tortuosus, the mountain jewel flower. It has these really pretty jewel flowers. And this species grows across latitudes and elevations in California. This is a picture of it at vegetative stage, and then this is a diagram of the flowers. And so because it grows across these elevations and latitudes, it has vast differences in seasonal conditions. And so the plants grow at different times of year in the same way that I described to you before. And so first, we know that climate is changing in all of these populations, but especially at high elevations. So let me break down this figure for you. This is a PCA that's describing variation in climate. So many different climate variables are in this. And so my PC1 explains 36% variation and goes from cold snow to warm no snow from the left to the right. And then PC2 explains about 25% variation and goes from dry to wet from bottom to top. And so each of these points are each of the populations included in my study, and their arrows point from historical to recent climate. And so you can see most of the arrows are pointing to the right, so towards a warmer climate with less snow. And all of them are mostly pointing upwards on PC2 towards a wetter climate, which might seem a little confusing at first, but this is mostly driven by higher precipitation in colder months. And so this is also representing a higher annual range in temperature as well. And so I want you to see that we're having climate change in both low and high elevation populations, but the low elevation arrows are shorter, so they're not as impacted by these changes. And then these two diamonds represent the 2 years that we grew the plants at our common garden site. And so 2024 represented a more normal climate year, whereas 2023 was a really wet year. I don't know if anybody was around in 2023 in California. It was a lot of precipitation. There we go. So for the methods, we performed a common garden experiment at high elevation. This is our site here. And we measured fitness of 23 populations from across the range. And we compared their home climate, so their most recent 30-year climate, to the garden climate that they actually grew in. And then this is a picture of the common garden there and some plants growing in the field. And so because I was interested in if we would see sort of seasonal dependent differences between low and high elevation plants and their performance at high elevation, I wanted to measure fitness at different points of the life cycle. So we germinated the plants in the greenhouse and we transplanted them to the field when they were 5 weeks old. We measured establishment 3 weeks later. And then we measured survival to budding in year 1 and fruit number in year 1. As well as first-year survival, which could occur from surviving as a vegetative plant or surviving to reproduction. Winter survival, survival to budding in year 2, and fruit number in year 2. And we had 2 metrics of reproductive fitness. The first is the probability of successfully making a fruit, so did you make any fruits or not? And the second one is the total fruit number. And then last bit of methods here is that we had 2 different metrics of climate distance. The first, temperature distance, so the home site's 30-year average minus the garden temperature they experienced. And then we had a more general climate distance that included temperature, precipitation, snowpack, and 12 other climatic variables. And so this one is also an absolute difference, so there's not directional, whereas temperature is a directional difference. Okay, so all of our results are going to look like this. We're going to have a fitness component on the y-axis and type of distance on the x. And so for temperature distance, anything that's negative means that the home site was colder than the garden, and anything that's positive means that the home site was warmer than the garden. So if we see a positive relationship, that means that sites from warmer home climates perform better. If we see a negative relationship, that means that sites from colder home sites perform better. And then if we see something that looks like this, that means we have a sign of local adaptation because we have the highest fitness at 0 climate distance. And then some of the figures are also going to have the general climate distance, and so this is just that the farther away from 0 it is, the home site is more generally different from the garden. And so here, positive relationship means that populations from home sites farther away climatically are better, and negative means that coming from a closer site is better. Okay, so what did we actually see? So here you're seeing temperature distance on the x-axis, year 1 survival on the y-axis, and our colors are the same as before, where our low elevations are warm colors and our high elevations are purples and blues. And what we see is something that looks a lot like this, where populations from warmer home sites had higher survival to the end of the first year than populations from more similar or colder home sites. And something else I want you to notice here is that this Wright's Lake 2, that's the local population. And so that had pretty high year 1 survival, but not as high as some of the low elevation plants we grew there. And then if we look at winter survival, here we have climate distance on the x-axis. And so here, again, we see that populations from farther climate survive winter better, and that's primarily driven by low elevation populations. And really surprisingly, our local population, Wrights Lake, had really low winter survival. I have some ideas about that if you have questions at the end. And then if we move to the second year and we look at survival to reproduction in their second year of life, here we have temperature distance on the x-axis, and now our relationship sort of flips, where now populations from either similar or slightly colder temperatures had higher survival to reproduction in the second year. And again, this is our local population, and it did quite well in the second year. And then if you kind of look overall at reproductive fitness, and so here I'm showing you a probability of making a fruit or not, and we look at that on temperature distance, we see that those effects from the first year and the first winter really have a big impact on whether you can actually make it to the point where you can make a fruit. And so we still have this positive relationship where populations from warmer home sites had a higher probability of reproducing than populations from more similar or colder sites. And so even though we saw a flip in the relationship in year 2, it was a little bit too little too late. Yeah, and again, that's our local population, not doing quite well. And then if we also look at total fruit number, this is including individuals that did not make any fruits. We still have this positive relationship, and this time with general climate distance, but it's not significant in this case. And you might see some different, well, okay, don't worry about it here. I'll come back to it later. Okay, so what did I find? So from this first part, if we ask our local high elevation plants showing signs of adaptational lag, we see that high elevation populations may be at risk of population decline because we see that they're not having great survival, especially through the first winter. And then if we look at this sort of seasonal dependence of will plants from hotter origins survive hotter summers better at high elevation, we do see that hotter origin plants survive first summer and winter better. But in the second year, we sort of saw a change where the high elevation plants were able to have higher survival. So there is still some variation going on here. And then if we think about assisted migration alone, so if we just took low elevation plants and moved them to high elevation, that might not be a successful strategy because those plants, especially in their second year of life, may not have the fitness, as much fitness as high elevation plants. And so instead, some sort of a strategic assisted gene flow may be a useful strategy here, where we take whatever genes help those low elevation plants survive the first summer and the first winter well, and combine those with whatever genes help the high elevation plants survive in their second year. And so now that brings me to the last question of if we actually make crosses between plants from hot and cool origins, do those perform better than, say, the local population by itself, or crosses just between low elevations and mid elevations, so just kind of different types of crosses. And so for this, we perform crosses between the local population and two populations from low, mid, and high elevations. And so if I bring back up the total reproductive output figure here to show you which populations we chose, we showed these two low elevation populations, these two mid elevation populations, and these two high elevation populations. And then we grew the F1s at the same high elevation common garden, and we compared the donor climate to the garden climate. And we measured the same fitness metrics. And so here, again, we have year 1 survival. This time we have climate distance of the donor population. So this is the non-local population that we crossed. And the colors are colored by the elevation of the donor. And then this is the local population by itself, and then all the triangles represent the F1s. And so what we see is a lot of variation here. One of our low, mid, and high elevation crosses all died, and then some of those had high fitness. And so there's a variation across the different types of crosses. And if we look at probability of reproducing, so making a fruit or not, we see the same variation where the same 3 crosses, one low, one mid, one high, had high probability of reproducing, and much higher than the local population. And then here, finally, is total reproductive outputs. And in this case, now we see sort of a separation happening between those F1s that were doing well before, where this is the mid-elevation donor actually had higher reproductive output than the other F1s and the local population. And so will crosses between plants of hotter and cooler origins have the highest fitness? Well, we actually found that gene flow from mid-elevations might be the best option in this case. And we also just saw a lot of variation, such that some populations may be better sources of gene flow than others. And so I think it's going to be really important when we're picking our sources for assisted gene flow to not just say, oh, we can just grab any low elevation or any mid elevation population. We have to be really careful about which ones we pick. And with that, I have lots of people to thank, my co-authors on this paper, my lab mates, and funding from the NSF, as well as the many undergraduate students that helped me out in the field to collect this data. And I will put up my research questions again and accept any questions.
Great, we have time for questions. If anyone would like to come up to the microphone, or I can bring it towards you.
Two things. Are there efforts to preserve the genetics of the really cold adapted populations and the mid populations as they are for the future in case, you know, the climate stabilizes and things get better, so that they wouldn't be maladapted at that point? And also, does hybridizing different populations cause the native pollination insects that are locally adapted to set each population to be less adapted to the hybrids?
Yeah, great questions. For the first question, since the species is currently very widespread and not at risk, there isn't currently any plans for conserving or even doing assisted gene flow actually, and this is all sort of testing theories at this point. But that is important that there is yearly variation, and so it could be that that first winter where we had really low survival of the high elevation populations was a warmer winter, and so maybe there was less consistent snowpack, and so maybe in a future winter where there's more consistent snowpack, the high elevation plants would actually do better. So I do think it's important that we're kind of balancing what we're doing with their conservation with what happens with our yearly variation in climate. And then for your second question, about like sort of the pollinators and how that interacts with the hybrids, we haven't seen any differences in sort of like seed sets. So once you're able to make a fruit, the fruit sizes at least look very similar. And we haven't done any sort of viability tests of those seeds yet, but I haven't visibly seen any differences with pollinators visiting the hybrids versus the non-hybrids, but it's a good question.
Thanks. I have a question about the hybrid experiment. Wouldn't you expect some improvements, some hybrid vigor in the F1 generation? And wouldn't you instead want to, or in addition, want to look at F2s and further on down the line?
Yeah, that's a great question. We also have been doing back crosses. Our final crosses that we're going to planting in the field are going to be back cross 2s. So this is sort of like the first peek at like what would happen in an early gene flow phase, but we'll get answers on more deep back crosses soon. Yeah.
We'll do one last quick question and then we'll move on to our third speaker.
I may have missed this, but are these obligate annual biennials? So in other words, they can't persist more than 2 years? Because this ties back to that reproductive schedule where you're talking about 1 year or 2 years. Well, what if they're growing more slowly at high elevations, then it will take them many years to reproduce? Did you factor that in, or are these actually obligate no more than 2 years?
So there are some rare plants at high elevation that grow more than 2 years. I can tell you in our experiment, we've only had one high elevation plant that's done more than 2 years. All the rest of them were obligate biennials. We because we left the plants up there to let them do whatever they wanted. So there is some variation in that, but most of them are obligate biennials. Yeah.
Thank you. Great. So next in our schedule, we have Dr. Sarah Bisbing here from the University of Nevada, Reno. And the title of her talk is Establishing Climate-Wise Seed Lot Selection for Sierra Nevada Reforestation.
Hello, a little more complicated today, I'm sorry. Thanks for having me. Thanks to Elsa and Jay and Ben for hosting me, because I am quite clearly the individual that is not like the others. And I'm surprised many of you didn't flee when you saw trees on the screen. I am suffering from a head cold, so I will do my best, but I may be a little off my game today, so I apologize for that in advance. I want to thank Brandy. She set the stage very beautifully for a lot of what I'm going to be talking about, so it saves me a little bit of time and trouble explaining some of the limitations of moving species or lots from low elevation and latitude up to high elevation or latitude. There's quite a bit of science on that in the conifer world, for those of you interested. I will also say, to start, the concept of assisted migration is not alarming or new in the forestry world, right? We've been planting and moving conifers forever. A good example, an endemic to coastal California, Monterey pine, Pinus radiata, occurs over something like 6,000 hectares in its native range in California and Baja, Mexico, and yet it's the most widely planted conifer globally. It makes up something like 90% of New Zealand and Australia's timber production. So what I'm going to talk about today is shifting from timber production-based reforestation efforts towards reforestation for the sake of keeping forests on the landscape. So that said, the concepts are very much in line with what we're talking about today. So local adaptation and plasticity and others. So for those of you that don't spend much time in forests, you have still likely seen images like this on the news or in driving across the state. Sadly, I, and now my son, see the world in dead trees, so I never miss an opportunity to take a picture or think about what's going on. In this slide are just images of some of the landscapes that I work in. For some friends in the audience, you can see Dawkins as a reference. He's my baseline for success everywhere. That's the dog. And then in the bottom is the coastal California Monterey pine population I was mentioning. So statewide, no conifer seems to have been saved from impacts across the state of California due to fire and drought and beetles. Just to summarize this really briefly, the map in the top right is a figure developed from aerial detection surveys of California. This is the peak of mortality in California following the 2012-2016 drought. The map shows 2014 to 2018 mortality. Between 2014 and 2020, the state lost around 168 million trees. You can see in the bottom right-hand schematic that cumulatively, up to last year, we lost close to 243 million. So it's non-trivial losses across ecosystems. We also know from science and observation that a lot of this is driven by an interaction between drought stress. In this paper, these images show climatic moisture deficit, so temperature and moisture interactions. And then having too many trees in the landscape, so too many straws to suck up the total available water to these species. Then following, we had the compounding stressor of native beetles. This pests and pathogens impacting already stressed trees and leading to this increasing mortality. You can see also that lower latitudes and lower elevations were the ecosystems that succumbed greatest to high levels of mortality. Additionally, we work in what we jokingly refer to as the pyro state. You know, California is a fire-adapted state. Nearly every ecosystem experiences fire on a regular interval. Very few are excluded from that. But the magnitude of impacts have changed pretty dramatically over the last decade. For reference, This total is not anomalous. Ecosystems burned at this acreage in the past. However, fires were smaller, more small fires, and lower severity, so less mortality from these fires given the way they were burning. Additionally, we have the threat of climate change, which I won't bore you with since we are all here for that reason. On the left just so it shows the relationship between dryness or aridity. It's classified in terms of vapor pressure deficit here with burning acreage across the western US. Additional papers have come out to show this is even a clearer relationship in the state of California. And then on the right, you see the vegetation-climate mismatch based on climatic effects. So this is ignoring things like fire and beetles, but can the vegetation that occurs in a given location persist into the future? The red is a severe mismatch. You can see it's primarily low elevation, low latitude, and the blue is relatively stable. We also know that natural recovery is improbable, particularly post-wildfire. We know this based on evidence on the landscape and based on the fact that we do not have available seed source and we don't have the right environment for establishment. In the furthest right panel, you can see a dotted oval around the Sierra Nevada, just emphasizing the systems that I'm working in, and it's particularly extreme there. With climate impacts, as well as fire and drought, we anticipate very limited recovery going forward without intervention. Where this gets me to is thinking about what we do in forestry and this idea of whether or not local as best. Sally Aitken and others have published a few papers on this, given that we have long moved trees and seedlots across the landscape and across the globe. But what we've done historically is what you see on the right. So this is a map that shows our seed zones. And in forestry, you have a state-based or regional seed zone map. So for example, when I want to plant in a given seed zone, I call the nursery or the seed bank and say, I'm looking to plant from seed zone 531 from elevation band 1,200 to 1,500 meters for ponderosa pine. I'm sent those seeds with the assumption that those local seeds will perform best. We already know and we already have evidence that that's not the case, and that's what we see in this image on the left. And this is modified from Sally Aitken and Bemel's paper. I think it's almost a decade ago now, which is crazy, but just illustrating this for conifers, this idea that local is best under the historical range of conditions, although the climatic niches will be shifting moving forward. And again, addressing the concepts that other speakers have presented on already. So this concept of assisted gene flow, so moving lots or provenances within a species range, which is very common, as well as assisted migration, so moving outside of the range. And again, this is not really a controversial topic in forestry, so just an example of where it's possible. What we're also working to do, in addition to planting and promoting climate-wise species and seedlots, is we are very much focusing on trying to ease transitions. So clearly, I love forests. I'm a forest ecologist, and that's what I've dedicated my career to. I also have a young son, and we love to recreate, and I want him to experience the world. So to me, I want to do a better job or promote a better job of making sure there are forests in the future. So our work is incredibly applied. And part of that is thinking about how can we ease these transitions? We know disturbance is inevitable. We know climate change is happening in unpredictable ways. So what can we do to make sure if my preferred species can't occur there, that we allow for other species, some sort of surrogate to keep forest ecosystems on the landscape? And that's what's argued in this paper by Connie Millar and Nate Stevenson here. So what you see in the bottom is just this idea of promoting species ahead of a disturbance happening, those that are likely to be more drought tolerant, or going in and planting those species so that you don't have this dip in services or issues through recovery. With that was born the experiment that I'm a PI on. It's called the Adaptive Management Experiment. It's a close collaboration with CAL FIRE and funded by CAL FIRE. Our 9 sites are on the right-hand side. It's largely a silvicultural experiment, meaning applying forest management techniques for climate change mitigation. And with that, we have 2 assumptions here. One is that climate change will occur in unpredictable ways, right? So we have to bet hedge or think about ways to strategize moving forward. And the second is that maladaptation is likely to occur if we continue with business as usual models. I won't go into the forestry details side of this, but there's a whole host of issues on the management side as well. For today, I'm going to focus solely on this transition in real time, so assisting maintaining forests ahead of time or post-disturbance in the hopes that we will maintain them on the landscape. On the left is our original experiment. It was 3 sites where we replicated different silvicultural treatments, all on CAL FIRE property or state property. Our plan was to embed these common gardens or provenance trials within these. And then as we were implementing these, so as we were going through timber harvest and planning for these different treatments, the 2020 fire season occurred. As part of that, one of our field sites had every acre burned on this landscape, and so we were forced to pivot. With that, unfortunately, and fortunately, I guess, we leveraged the 2020 and 2021 fire seasons to bolster up to 9 field sites. These span about 4 degrees of latitude in the Sierra Nevada from north to south, and they cover west to east. Within these is the context of what I'm talking about today for reforestation efforts. So we got to work, and we got to planting, which for research scientists is definitely not what we are adapted to. So we did our best, and in the end, we were pretty successful. What you're looking at here on the top is just the 5 species we planted. We planted the 4 most commonly reforested conifers in the Sierra Nevada, ponderosa pine, Jeffrey pine, sugar pine, and incense cedar. And then we added in giant sequoia as our assisted migration trial species, and because it is beloved. People want to see giant sequoia on the landscape. Ultimately, we're aiming to develop these tolerance curves with conifers. They very clearly, in general, follow this bell curve that presumes local adaptation, and so we're working within this framework. On the right is just the lots that were selected. The gray triangles, the gray X's represent the available lots we had to pull from. Each color represents a different species. I won't go into much detail here, but if you look, each species is represented across the entire Sierra Nevada, with the exception of giant sequoia. Giant sequoia, which is Sigi on here, is only in the bottom half because that's within its natural range. It was planted outside of that. What you see here is a clearer map overlaying our field sites on top of the seed lot selection. The triangles represent where we planted, the seed lots represent the distribution. And then we did something a little different than what typically happens in common garden studies. So I am an advocate and I've been a practitioner of the local versus foreign gardens. We didn't do this here. We selected based on climatic variability of minimum coldest month temperature, so winter extremes. There's a huge amount of literature on conifers to suggest that's a limiting factor to growth and survival, particularly in these high elevation environments, and particularly in the Sierra Nevada, where we have these variable snowpacks and variable temperatures. What you see here is just the distribution of CVs in general. Jeffrey pine and ponderosa are widespread species, so JP and PP. Jeffrey pine has the largest breadth that we were able to select from, particularly at upper elevation. But we selected the 6 populations from that lot, the range of lots that had lowest variability in winter temperatures, and then the 6 that had highest variability, under the assumption that populations that have experienced high variability climatic conditions over their history are more likely to tolerate the extremes of the future. Just for your awareness, you can see how this is arrayed in climate space. I don't love mean annual precipitation or temperature. I think they're not very valid climatic predictors, but the scientific audience loves it. And so you can see here how these are compared in space. We don't cover the coldest temperatures very well, just based on the forest type we were planting in, the Sierra mixed conifer, but we do cover the range of precipitation conditions there. In the end, what we have planted is the 60 total lots, so 12 lots per species, across 29 total gardens, covers about 17 hectares for scale, and we've been tracking roughly 46,000 individuals across the range of the Sierra Nevada for the last 5 years. These are just images of the planting scheme in the top, where each individual tree was flagged prior to planting, and the bottom, some drone images that just showed what the gardens look like. A really quick schematic, because we've been tracking survival over the course of a field season, so our crews visit these gardens every month over a 6-month period, and then over years, we're able to track mortality over time. Our next step in this study is to look at these climatic windows that are specifically influencing survival or mortality. But where we are right now is in sort of the early summarization stage. What you're looking at here is just the distribution of our field sites. I promise you there are 9, but they were planted over 2 different years. Some sites, highlighted in blue, we planted for 2 years in a row. And what I like to see here is that there's consistency in how species respond. It's not perfect, but it's relatively close in who survives and who is less successful. You can also see that the blue Jeffrey pine is the most successful. It is our generalist species. Additionally, what's really interesting, this Esham site is the lowest latitude, lowest elevation site. It is our in situ climate change site. We planted there intentionally post-drought mortality to see if conifers could be reforested. You can see that it's not very likely. As of this last year, I think we have 1 Jeffrey pine surviving out of 3,000 trees planted. So we are not maintaining the transition here. It might be a system we walk away from. What this looks like generally, and I just have a few slides left, is thinking about these distributions. The more interesting patterns so far are at the species level. We're in the process of developing Bayesian models that look at these time steps for each individual lot and over time, but I'm going to focus very generally on the patterns at species level. Brandy already did a great job of explaining transfer distances, and here you can see for climatic moisture deficit, anything to the left of the 0 is less climatic moisture deficit, so less drought, less stress. Anything to the right is more stress. The general things we see are our 2 generalists, so ponderosa pine and Jeffrey pine. As one would expect, they cover a wider range of environments. They are a dominant species, both in the Sierra Nevada mixed conifer, and they're able to tolerate a wider range of conditions. But then we also have these specialist species, so less success, more narrow widths in terms of their confidence bands. When we move in thinking about minimum coldest month temperature, which is how we selected our lots, we see some more interesting patterns in terms of response and survival. We see that a few of our species that are more narrowly distributed, so incense cedar and sugar pine and giant sequoia, so the 3 green, yellow, and orange in the middle, have an optimum in their home environment and more narrow range of tolerance, right? We don't need to be moving these to higher elevation necessarily because they won't survive. It's not an effective use of your reforestation efforts. Additionally, when we look at beginning of the frost-free period, so again, going back to what Brandy was talking about in terms of moving lots in these different growing seasons, the left is earlier growing seasons, so longer seasons. The right is shorter growing, or I can't actually see from here without my glasses, but hopefully you can see it because it's an odd angle. But we have specialists again, and we have generalists, and Jeffrey pine is able to tolerate, again, this wider range, where our more endemic, for lack of a better word, are more narrowly distributed here. This is where things get, unfortunately, a little messy and are currently not following our predictions, but what we looked at was we ordered our populations or our lots by high CV versus low CV to test our hypotheses. In general, for drought stress, provenances from higher variability climates are more plastic. They can tolerate a wider range and are more drought tolerant, but as one would expect, less drought is preferred across all species, are the general patterns here. And these are things we'll be refining in the coming months because they don't look the way I expected them to. Next, when we're looking at minimum coldest month temperature, there's a huge amount of within and among species variation. It's very much based on the species' adaptations and the lot level adaptations, and this is one we need to spend more time parsing out. And then finally, the frost-free period. So generally, most species are preferring this longer growing season over time. So some takeaway messages, particularly because I'm sure I'm over time, is going back to some of the general patterns and general concepts we think about in adaptation is plasticity is expected and widespread in these conifers, and unlike annual and biennial species, you have a little more time with these species to get responses. In general, in my experience, conifers take about 3 years or so before they start to be really impacted by drought stress, which leads to mortality. We also do see strong local adaptation in more narrowly distributed species. and that these populations or species are more plastic to drought than frost, which is unsurprising in this Mediterranean climate where we have very droughty summers. The winter, which is highly variable in snow and temperature, is more impactful on who can survive. So all of that is to say, we should not be moving trees higher in elevation or latitude necessarily without a lot of critical thought around that. The final takeaways are thinking about bed hedging, so making sure you're planting local and non-local seed sources in reforestation efforts, so more diversity in lots, planting generalists, and then making sure that there's a wide range of variation within and among species here. And I probably do not have time for questions, but I can take them later.
Thank you, Sarah, for that excellent talk. Indeed, we are a little over time, so if you have questions, I encourage you to save them for the panel, which will come after our last speaker. So it's my great pleasure to introduce Dr. Julia Michaels, who is here from Hedgerow Farms. And the title of her talk is Native Seed Producers Can Play a Proactive Role in Conservation Genetics.
Okay, this is like the biggest room I've ever spoken to. This is, I mean, just size-wise. Which one do I do to go forward? The big green button. Duh, yeah, sorry. Hi, everyone. I'm Julia Michaels. I'm a restoration ecologist, and I work at Hedgerow Farms, which is a native seed farm located right outside Sacramento in Winters. And thank you so much to the organizers for inviting me to be a part of your panel. It's a really interesting topic. I think we really need to be thinking in these, ways that are creative and sometimes rub people the wrong way, right? Local is best is a very strong paradigm right now, and there's some really interesting research coming out about adapted gene flow. Okay, so when I was in grad school, I was introduced to Hedgerow Farms as the University of Hedgerow Farms, and that's because there was just so much learning going on as we sort of built this, this whole world of seed collection and growing and harvesting, and obviously people have been doing this for millennia, right? So I'm not saying that we invented the idea of seed farms, but just turning it into something that's actually like an industry was happening, and how do you get different plants to germinate and grow in these agricultural contexts? So anyway, Hedgerow Farms has always had a really special place in my heart, and so when I was teaching at Reed College, Restoration Ecology, and a job came up, it was just obvious to me to come take it. So I guess that makes me academish, because I get to basically collaborate with these fantastic researchers, but I'm mostly doing more like the public outreach and education, and trying to help make these projects a reality. We'll talk more about that in a moment. So again, Hedgerow Farms has been around for over 35 years now. It's about 300 acres, and we specialize in collecting and growing local ecotypes, locally adapted versions of the plants that we produce. We do custom seed collection and production, so if you're interested in getting a local ecotype more available, I'd love to talk to you about that. And we do a lot of technical assistance and consultation. Even though we're not officially consultants, we do a lot of talking through these types of decisions with people. So here's our farm. It's gorgeous. If you're ever in the area, please come. You can see that yellow field is a little sparse. That's because it's a G1 field, which means we collected the seed, and then that was the first time we'd ever planted it. So over time, it fills up. So we grow, hundreds of different types of species, 50 at a time, more or less. And this is a great photo taken by a middle school teacher and his students of all the different seeds that are in our Central Valley pollinator mix. So you can just see all the different sizes and shapes, and it's just really cool to think about seeds and all their different adaptations, and it's a really fun job. So how do we get the seed? We go out into the wild with permission, and we collect small amounts. There are people in this room who do that seed collection for us. We have an incredible crew that's been working with us for pretty much their whole careers. Sometimes I get to go out, not very often, but I sure as heck am going to document it when I do it. So there's me. And then we plant that seed at the farm using agricultural techniques, combines and tractors and all of that, we plant them in rows. It's really interesting to see native plants growing in these big fields, right? It's not what you're really used to seeing. And then we harvest that. Usually, since they're annuals, we combine the whole thing, or if they're perennials, we'll just harvest off the top. And then we clean that seed using equipment that was built for cleaning barley. And we've managed to sort of turn it into the type of machinery that we need to clean these hundreds of different types of seeds of different shapes and sizes. So, that's what we do. So Hedgerow Farms is actually part of a larger company called the Native Seed Group. We have 4 locations to our farms. So we actually have a Hedgerow Farms South. If you are near Los Alamos or Santa Barbara, you can come visit that farm. And then we have our 2 distribution locations. And across those locations, we actually have 570 acres in production, 600 plus, probably more like 800 at this point, species available, and 1.7 million pounds of seed in storage. Not talking about, our rare forbs. A lot of that is big workhorse species, right? Like Elamus glaucus and Bromus, things like that. But we've got a lot of seed, and it's being used and deployed across restoration projects all across the state. One of the things we really are trying to work on is making sure that we keep all of our seed of known genetic origin, meaning we know exactly where we collected it and how long we've been growing it for. And I'm just going to talk to you a little bit about sort of what that means. So again, we have all these different ecotypes. We go out and we collect, we store them, and then we try to not keep inventory on the shelf for too long because seed doesn't last forever. So I'm not saying you could go and get any one of these ecotypes at any moment from us, but these are like the stock seed that we have available to plant. So, these are just some examples of all the different places we've collected. And, when you think about this, though, it's not like we have an ecotype of every single species for every single location. So when you're doing restoration, you have to make choices. And so that's something that we have to do all the time, is help people make choices about where their seed provenance is coming from. Uh-oh, am I frozen? Did it lose battery? Oh, back. Yeah, there we go. Oh yeah, all right, let's just go here. So, why do I have a California condor on my slide? Because the more you think about it, like what we're really trying to do is take seed from the wild and keep it as wild as possible and then return it back, right? We're basically captive breeding for plants. And that's my dad helping me collect some Clarkia. So, what we're really trying to do is make sure that we have genetically appropriate material available for the scientists to decide what to do with. And that's a lot harder than it seems. So, first of all, one of the things we've been doing is working with the California Crop Improvement Association, and they are an organization that helps, vegetable farmers, but they have a whole native seed program where they basically will come out and they'll certify. They'll say, yep, this seed that you collected is from where you said it was from. And that's called source identification. So we're trying to improve the use of this service, basically, so you can get an actual tag on your bag that says, the seed came from where you're saying it's coming from. Otherwise, the customer is just kind of trusting us, right? And when you think about it, we're never going to be able to source identify every single lot. There's a lot of work to do. But the key ones that, the conservationists that we work with tell us are important, we can do that. And then once we get that seed to our farm, we have to think about genetic isolation, because if we're growing, 5 different ecotypes of Stipropulchra, they have to be spread out and far enough from one another that they're not cross-contaminating. Luckily, at Hedgerow Farms, at least, we are a mile from north to south, so we actually have the ability to do some pretty good distance. And we are using the best available science to do that genetic isolation, and the California Crop Improvement Association has standards that we follow. And so that's them coming out, checking the fields, making sure we're doing the best practices. The other thing we have to think about is generation since collection, right? So, you collect a local ecotype, but if you've grown it for many generations at the farm, does it start to domesticate? So there's been, concern about that. So there was a paper that came out, I think, in 2022 about plants that have been collected and cultivated for ecosystem restoration, and how they might start to evolve towards a domestication syndrome. This was done in Europe with, I think it was like 19 different plants, and not all of them did this, but there's concern that things like if you are harvesting a field all at the same time, it's going to start selecting for just those seed heads that came up at that time, and also for seed heads that didn't shatter, right? Because if they drop to the ground, that's what plants are supposed to do. They don't get harvested. So over time, with annuals especially, you have to be concerned about generation. And then, of course, ecotype, which we've talked about, and this is, we've known about ecotypes for a long time. This is a paper from like 1940 or something. But, the idea that local is best is something that is, really being debated right now. And as a seed mix designer for hedgerow farms, I spend a lot of time talking with people about which ecotypes to choose. The California Native Seed Strategy, there's a whole session on it tomorrow, but they're trying to figure out seed transfer zones. And in the Intermountain West, we have defined seed transfer zones. You can move a seed around within this zone, but you can't move it outside. This map in the bottom left kind of shows suggested seed transfer zones, and of course the farther out west you get, the more complicated they get, because there's so many microclimates in California, and I don't know if we'll ever have that same type of seed transfer zone. But it's really something that we have to decide as a conservation community what we're comfortable with. So what's my job? Now as I'm academic, I do my best to provide science-informed guidance to customers. Here are like demands that I get from customers all the time. So they'll say, we want coastal counties only. I want my seed only from a coastal county. Doesn't matter if it's, San Diego coastal or Humboldt coastal. I want coastal. And others will say, okay, I only want seed from my county, right? My project requires Santa Clara County seed only. San Mateo will not do, even if it's 5 miles away. 50 miles from the project site. That's one that we see quite often. Actually, we're working with a client right now that is only accepting seed collected from 5 miles from the project site. The more specific you get as a client, the more expensive the seed becomes, right? So lower generation seed, more expensive, and the more local, more expensive, because we have to do all of this scouting, and we have to even find the seed. Sometimes it's not even there, and it's really an ask. So you have to think about why, right? Why do you want local seed? But actually, really, like the question I most often get is, what is an ecotype? Because we're all thinking about this all the time, but there's this whole world of restoration that's happening without us guiding it. All of the mitigation firms and all of the agencies that are not ecologically minded, et cetera. So I do a lot of explaining, what is an ecotype. And I can go into different papers that have shown, but this whole session has been about how, local is best is, from a do no harm perspective, perhaps it's the right approach to take, but we're finding more and more that these regional admixtures may be more resilient to invasion, to drought, et cetera. So, these are conversations I have with people. Do you really, really want to spend all your money making sure it's collected from 5 miles of the project site, or are you interested in trying to create something, a little more flexible? Okay, so we have 3 minutes left, according to me. I want to just sort of ask you guys, if you're on a limited budget, which species would we prioritize for the most local ecotypes and the low generation seed, right? So here's a mix that we sell all the time. Bromus citrensis carinatus, Almus glaucus, Festuca microstatis, Trifolium, Stipa, lupin. So, looking at this, we don't have time to do this as an exercise, but think to yourself, which ones would be the ones that you would think are the most important for a local ecotype? And I'll talk you through my thought process really fast. So I have a little flow chart that I kind of use. So first I think about the breeding system. So is the plant wind pollinated? Is it self-pollinating? Is it does it have polyploidy? Is it diploid? So if it's something fancy, maybe that is a tip-off that you want to try and use a more local ecotype. Does it have specialist pollinators? If it has a really specialist pollinator, maybe you don't want to introduce a ecotype that blooms at a really different time, right? So just another thing to consider. What are the restoration site characteristics? So just because it's, local, maybe if it's a road cut, it's a really dry site, or maybe the soil is really different, or is there a remnant population of the same plant that you would be flooding with these new genetics, in which case you'd really want to be doing intentional, assisted gene flow. If it's not even present on the site, maybe you're not as worried about conserving those genetics. And then, finally, what are the alternate ecotypes available? So, maybe if there's absolutely nothing that is appropriate, that's the time to spend your budget on collecting and growing with us. So that's kind of what I go through. But again, as a seed company, we're doing this every day. And I think it's really great that we're sitting here and thinking really carefully. And at the same time, we have to recognize that we don't want the perfect to be the enemy of the good, or whatever that saying is, right? So the more we can communicate between the thought leaders, the academics, and the people who are, designing seed mixes on their phone as they're, riding the bus, like myself, the better we can be, because I don't want to be sloppy, and I also, need to be making these recommendations all the time. So that's why I'm just so glad to be part of this. And so yeah, there's just some pretty pictures of that. And then I just want to remind everybody that Hedgerow Farms is a place that has always been a venue and a hub for native plant conservation and restoration, and to me it will always be the University of Hedgerow Farms. And so, if you're ever in the area in winters in Sacramento, I encourage you to come out and visit. It's really a special place, and my contact information is right there. There's a QR code to our website. So yeah, that's what I have to say.
Okay, so
wow, that was awesome. So I'm involved with a nonprofit in the South Bay down near Los Angeles, and so much of what you've talked about is just making me think a lot. Is there a working group that I could get plugged in with this? Because, okay, we'll talk.
Yeah.
So where we're at, there's an endangered butterfly, the Elsa blue butterfly, for example, and its host plant is Eriogonum parvifolium. So we are hypersensitive with that one. We make sure we're only getting the buckwheat that's from seeds grown locally. But I did, for example, want to have a South Bay super bloom, so we bought a whole bunch of the coast poppy from one of the nurseries in your group here. And it turns out, and contaminate's a strong word, right? But there's poppies that are not that.
Right.
And so now the question becomes, do we spend effort to rip out the ones that aren't the coast poppy, right? And so, anyway, I'm throwing that out here because to me this is such a critical question, where to spend our efforts. And anyway, so
Yeah.
thank you. I don't want to get plugged in. And do you have thoughts on, for example, the coast poppy, for example?
Well, before my time, and I'm sure someone in this room can better answer this, there's this whole story of the Caltrans poppies. Raise your hand if you're familiar with the Caltrans poppy thing. Okay, so, right, so this is the scary example of Caltrans bought a bunch of poppy from SoCal and then spread it all across the state, and then the Southern California version did so much better in Northern California, and it took over and depleted the genetics. And I'm not an expert on this, so maybe talk to some of those people that raise your hands. But I don't know. I think it's so subjective, right? And yeah, I wouldn't spend a bunch of time ripping out poppy. That seems like I would rip out some invasive ice plant while I was at it. Anyone else?
Yeah. So I have a question about the number of genetics to use for, or genetics from the native population from wildland collection. My organization, we have a small seed bulking operation to the point where we've been able to use all wildland collections, but we're getting to the point where we're debating reusing previously bulk seed. At what generation from wildland collected to grown in the bulking field do you say that seed is no longer viable for bulking?
I was literally sitting at the farm on Monday talking to the farmer, being like, all right, where are we going to cut it off? Because we're really sensitive to this now, right? there's no right answer for any species. There are trendy things. I hear G3 or below often. I don't know if that's, people are looking for clarity, so I feel comfortable with that. I don't know. If it's a perennial, right, you can have it growing for 6 years and then it's not, it's still G1, right? Versus annuals can maybe adapt more quickly. I wouldn't feel comfortable selling an annual that's like D10, something like that. That I know for sure, right? Beyond that, it's up to you all researchers to tell us. Yeah.
For questions for Julia, but we're going to turn over now to have a panel discussion with all of our speakers, so please stick around, but let's have a round of applause for all the speakers in the session.
Okay, all right, we're back on. Thanks, everyone. Thanks to the great panel for those talks. We want to spend most of the time of this giving opportunity for you to give questions. So get your questions ready. We're going to be interrogating the panel here a little bit. This is a really eyebrow-raising topic we found over the years, and a lot of it comes down to clarity, just understanding what people mean by assisted gene flow, and also, we learned a lot about how it can be beneficial in some of the experiments, but, potentially, hopefully, little variation is lost. We need to be strategic. I think that came out in our talks. Variability and plasticity are really important factors that we need to think about. And the logistics can be super complex. So we want to start with just one general question for the panel, which is, we've heard a lot about different aspects, themes, and findings for assisted gene flow today. What do you see as the biggest gap between what we know scientifically and what land managers actually need to implement right now? So you could all address it if you want, and then think about your questions.
All right, I'll go first on this. I've thought a little bit about this at different times. I think that what most land managers want is they want a very specific set of guidelines that are general across different species that they can just implement and use as a justification for doing some assisted gene flow or doing some bed hedging. So for instance, they want to know that you should be planting 5% of the natural population that's there. And if you do that, you're not going to lose any native biodiversity that's there. They want to know that you should be planting seeds, or they want to know that you should be planting seedlings. They want to know if it's 100 miles away or 50 miles away, or how much we should be bet hedging So what proportion of our seed should we actually be moving in? So I think it's those logistical things that land managers want to know that we haven't been providing yet, and that we need to be providing both from a simulation perspective and from an imperial perspective in populations.
I think you nailed it. I have nothing to add.
Yeah, I guess I'll just add that something I've been noticing with our species is that there's just a lot of variation year to year in the climate, especially at high elevations in the Sierra Nevada. And I think that makes it harder to give simple answers, which is, the easier thing for land managers. Oh, you're ready.
I have one to keep you safe. I think my perspective is a little different based on what system I work in. Not completely, but in forestry, the action is to reforest after a disturbance, whether it's man-made or not, right? And we're not very patient, particularly in the Sierra Nevada, right? Much science has shown that, and I'm sorry because you all study the herbaceous and shrub species that I'm going to comment on now, but to foresters, that is competing vegetation. And if you want to forest there, you want to promote the trees. So it's a very different lens when you're talking to forest managers. And it's not that they ignore the other aspects. A true forester that is a professional thinks about every aspect that they're working with, but their priority is to make sure that there are trees long term, and the key is early establishment. So we're a little less patient. Forestry is also very prescriptive. We write what's called a prescription, and when you go out, a forester wants to know exactly what to do. So I find a lot of impatience with that too. It's very unsatisfying for the foresters I work with to hear me say, well, I would like you to continue to plant some local populations. And I think we also need to consider having a diversity of species, a diversity of seed lots, right? And think about building resilience into that system. And by resilience, I'm going to define it a little differently, but having something persist, right? Or something recover, even if it's not your desired or your local. So the bet hedging idea is a critical one, and the portfolio effect is critical, but it's going to be very hard to tell a manager, like, move from these locations. It's far more, I want you to think critically about the range of climate conditions you're selecting seed sources from, which is also challenging and hard to apply. So I think it's a long game strategy, but the first step is this bet hedging and portfolio effect.
Great. Let's hear from the audience with questions. You can ask generally or target certain speakers as you wish.
Thanks, Ben. Thanks for the good talks. I just wanted to be sure that the cautions and potential dangers get their due here. The famous ecological principle, the precautionary principle, I think it's really important that given how different every different situation is, that we have the careful research and we really know what we're doing before we intervene. Because, as we all know, plants go in for this kind of local variation structure, sometimes within the north and south sides of one valley can have, different genotypes. And, there's a real danger to homogenize. This idea of portfolio, personally to me, is scary, because the idea is you don't want to mix every kind of adaptation that species has in one place. It's the local variation that's part of the evolutionary process. So my question, more broadly, we're not interested in just having plants preserved. We're interested in having natural communities. This gentleman here mentioned butterflies and things. So I was wondering, in particular, on your list of the criteria for what to select for restoration, I didn't see in there a list of what was the community or what's the expected community there. Because for insects and fungi and things, so I think we're trying to preserve ecological communities and evolutionary and ecological processes, not just taxa, which we can do in a garden. So I'd just like to know what you guys think about the potential downsides of the assisted migration or assisted gene flow.
Yeah, I think that's kind of the default, right? Everybody is afraid of being the next Caltrans poppy situation, or causing an endangered butterfly to go extinct locally or whatever it is. I definitely think that that's a really good point. When I think about restoration site characteristics, I'm thinking also about whether there's already a population of this plant that we're seeding there and what it would do to introduce that seed. The problem is we just don't always have that information. So I think it's helpful to know the questions to ask the practitioners that are coming to us. So I'll definitely keep that in mind.
I'd like to address the other side of that, though, too. So while we can cause harm by doing something, we can also cause harm by doing nothing, right? So if you have populations that are already going extinct, if you have decreases in gene flow because you have inbreeding depression, you have lower numbers of individuals, we have to be able to let the evolutionary process play things out. One thing that I haven't heard very much of here is that if we are introducing a bunch of different varieties into a population, what's going to happen to the ones that don't have pollinators there? Well, over the course of generations, they're going to die out of the population. And yes, you may have wasted some of your money if you're paying somebody to breed different seed varieties for you, but we have to let the process of evolution be able to play out within these different populations too. The question is, what's the right mix of this? But there's certainly a problem with doing nothing as well too, and there's some, we have some risk management that we have to go from there too.
Yeah, I have a lot of things I could say about that, but I completely agree, and I think caution needs to be made in anything that we're doing. I want to echo what Nick's saying in that in my world in forests, where we've obviously made a whole host of poor decisions, and I came into forestry as a raging hippie that wanted to hug and climb trees. I thought Julia Butterfly was like my role model in life. I learned over my career, however, exactly what Nick is saying, that in many cases, doing nothing in systems that are dynamic and constantly changing is the wrong choice. And we see that all over California with poor choices in removing our natural disturbance regimes and then not adding a surrogate for it. And a lot of what we're seeing, and I know this is a different audience, so I'll just clarify it, but it's not just climate change. We have made a whole host of very crummy decisions, particularly in forest management, that is created a really unstable foundation upon which climate change is overlaid. And so we have already made bad decisions and then done nothing for a long time. So I agree with Nick, the strategy is not to only plant a variety of things, but you also need to think very pragmatically and very much about your objectives. And I personally am going to be super bummed if there are no forests on the landscape because we made really poor choices and trees can't persist on the landscape. So I think we need to balance those two and think very carefully about the actions we take, but I don't think sitting on our laurels is the right choice, frankly.
I'll just add briefly that I think some of Nick's work showed that sometimes when you do assisted gene flow, it's not having these big impacts on swamping out the local genetic variation, and so I feel like that's one of the main concerns. And so I feel like, doing crosses or introducing the gene flow can help maintain the genes from the local population, but help them out. So maybe that helps with some of the scariness.
And maybe to start with, just thinking about, the silos that we all think in and remembering how many people are doing restoration every single day and adding seed to the landscape. Maybe the thing we can at least all agree on is what we really don't want to do, right? Maybe there's a few principles that would make, everyone feel comfortable. Like maybe you always start small. Like maybe you don't just start doing, thousands of acres, right? Like that seems like a good place to start.
Great. Other questions out there? We've got.
Yeah, I was wondering if prior to doing assisted gene flow, the populations you were adding new individuals in from elsewhere, if you preserved seed from as much of the genetic diversity as existed prior to adding new individuals in as possible, be it trees or any other kind of plant. That way, not only you preserve its initial genetic state for in case things change back to the way they were, but also to help in genome sequencing studies to help you reconstruct ancestral genomic states of species ancestors?
Yes. I think it's really important to preserve all the seed that we can over time. And you never know, that threatened population, that seed set, and that diversity that's there, that might be useful in future points at different places throughout the range as well, too. So don't think of it just as a backstop plan, but it's also a useful source for possibly other populations, too.
Yeah, and I also just say, like, a lot of plants have natural seed banks, and so even if we do assisted gene flow, seeds from the seed bank can germinate in a future year and reintroduce some of that old genetic variation. So plants already kind of have a strategy for that built in.
And there's some new funding for seed banking that was recently passed, and I can't remember what it's called, but it was the California Institute for Biodiversity got it passed. So we should have an unprecedented opportunity to be seed banking this next few years.
Terrific. Are there questions out there?
Hello. Yeah, I have a more like tree forest related question, but I'd like to hear other people respond. If there is like a significant mismatch between the climate and like the forest regeneration, should it not be a forest anymore? Like, should it become the next succession in that, like a chaparral bushland or like a woodland? And then maybe like the forest could move into higher latitudes, or like, what do you think about something like that?
Oh yeah, I don't have opinions on anything, but on this one, yeah, no, I mean, I agree. I only briefly talked on it because my brain is not highly functioning today. I was taking too long, but in our in situ climate change sites in the lower latitudes and lower elevations in the Sierra Nevada, I do think that's what we need to do. We do need to be walking away from sites and not investing, you know, from a resource perspective, time and money and effort into that, but also from a successional processes and what should occur there, right? So those are ones we walk away from. Where I'm more concerned is I work at higher elevations, so I also work in whitebark pine, I work in giant sequoia, old growth giant sequoia and redwood, and really beloved species. In those systems that will remain forest, like we see post-fire regeneration in these old-growth giant sequoia forests, but it's not necessarily what we want to see. I think in many cases, we do want to maintain forest where there will be forest. And my view has changed over the years that I am less species-focused, even though that's what we study, because it's critical to maintain ecosystem services and human services, right? Tree cover for mountain biking, tree cover for particular wildlife species, whatever we're using it for. I do have a lot of concerns about high elevation and high latitude. As a forest ecologist that studies these systems, if any of you have tried to plant seeds or seedlings in high elevation systems, it's nearly impossible. One is most of that seed is cached, right, by a disperser, so birds or rodents and other species, bears. They're put in sites where only some of them germinate, right? And so we've done studies where we're using drills to try to get seed of whitebark in the ground. And it's the probability of success at that elevation, particularly for species that don't have those adaptations to drill in, is really limited. And so I think we're going to see constraints on both. We are seeing, I don't know if my colleague Hugh Safford is in the room, he was earlier, but he has been tracking Jeffrey pine at high elevations, and he has found it upwards of like 4,000 or 5,000 meters. And so we are seeing the Clark's nutcracker cache higher of other high quality seed, so there are potential species that will move, but it's going to be really limited. So really where I work is in that moderate mid-elevation band where we have forest, we expect there to be forest, and we want to maintain some of those. So I agree on all your points. My only comment would be to have some concern on those higher latitude, higher elevation ones. Success is pretty limited there.
All right, well, 20 minutes goes pretty fast. So we're pretty much at the end there. I would say reach out to these incredible folks for tips on future resources if you're curious about this topic, like to learn more. But let's thank our panel. That was really great. Thank you.